Dot plots act as a visual representation of the CBC, with each dot representing a cell that the analyzer counted. They serve as two primary purposes. One, quality assurance on how the analyzer performed for each sample, thus creating confidence in the reported results. And two, as a visual tool to help identify red blood cell, white blood cell, and platelet morphologic abnormalities. In the red blood cell dot plot, more than 30,000 digitized events, each representing the signal for an individual cellular event, are presented for evaluation. Forward light scatter data, which is indicative of cellular size, is presented on the y-axis. Larger cells are located at the top of the graph, and smaller cells are located at the bottom of the graph. The fluorescent dye used by the Procyte is highly sensitive and specific for nucleic acid, RNA, and DNA. In the presence of laser light, cells that absorb this dye fluoresce. Fluorescent signals for these cells are represented on the x-axis. Those cells with greater nucleic acid, RNA in the case of red blood cells and platelets, are located left to right along the x-axis. Mature red blood cells are represented as red dots. These cells have no cytoplasmic RNA and are therefore located on the left side of the x-axis. Reticulocytes are immature red blood cells, and because they still have cytoplasmic RNA for hemoglobin production, they are located to the right of the mature red blood cells. These are the magenta dots. These cells circulate in the blood for a maximum of 2 to 3 days, and as they transition to the mature red blood cell, they're found closer to the red dots. Platelets are marked as blue dots. These are typically much smaller than the red blood cells and are located near the bottom of the y-axis. Larger platelets with more cytoplasm containing RNA are found higher on the y-axis and further to the right on the x-axis. In the white blood cell dot plot, 10 to 15,000 digitized events, each representing an individual cell, are presented for evaluation. On this dot plot, the optical fluorescent signals for individual cells are represented on the y-axis. The same fluorescent dye, which is highly sensitive and specific for nucleic acids, used on the RBC run, is used for the WBC run. Dots represent cells with increasing cellular nucleic acid, RNA and DNA in the case of white blood cells, as they are located bottom to top on y-axis. On the x-axis, the sidelight scatter data, which correlates to cytoplasmic complexity or granularity of the cells, is presented. Cells with increasing cytoplasmic complexity will be located further to the right on the x-axis. The separation between cells on the fluorescent scale, y-axis, is primarily related to differences in cytoplasmic RNA content. The neutrophils, represented with purple dots, are end-stage cells with no ability to divide or produce proteins, and therefore they have little to no cytoplasmic RNA, and they're located near the bottom of the dot plot. The eosinophils, represented by green dots, are similar to the neutrophil regarding their maturation and cytoplasmic RNA content, and in the case of the dog, they're low on the y-axis. However, because of the granularity of the cytoplasm, they're located to the right of the neutrophil. Each species has specific eosinophil morphologic features and will be located at different positions on the dot plot. In the case of the cat, eosinophils are located above and to the right of the neutrophil. The basophils in most mammalian species appear slightly less mature than the neutrophil and will have slightly greater amounts of cytoplasmic RNA and are therefore located above the level of the neutrophil. Each species has specific basophil morphologic features and will be located at different positions on the dot plot. The canine basophil depicted here typically has very few cytoplasmic granules and is therefore located above or slightly to the right of the level of the neutrophil on the x-axis. In the case of the cat, basophils are above and to the right of the neutrophil cluster. The lymphocytes may have significant variability in morphology and therefore are typically dispersed over a wider area than the extremely uniform granulocyte, neutrophils, eosinophils, and basophils populations in most species. These cells have higher amounts of cytoplasmic RNA and therefore are located higher on the y-axis compared to the various granulocytes. Since they typically have no significant granularity or cytoplasmic complexity, they are located similarly to the neutrophils on the x-axis. With increasing reactivity, there is commonly more cytoplasm and more cytoplasmic RNA. These cells will be located higher on the y-axis compared to the normal lymphocyte population. Monocytes are the least differentiated white blood cell in circulation and have the ability for cell division and marked protein synthesis, requiring abundant cytoplasmic RNA as these cells migrate to the tissues and differentiate to macrophages. Therefore, these cells are located highest on the y-axis among the normal white blood cells typically found in circulation. As they become activated, increased amounts of cytoplasmic RNA may be seen, and digitized events may even be found bordering the very top edge of the dot plot scale. Activated monocytes and other reactive or immature cells may be found here. The orange population consists of unlysed red blood cells and fragments. Because the red blood cells have no nuclear content and no cytoplasmic RNA, they have essentially no fluorescent light scatter and fall lower on the dot plot than any white blood cells. These are the basics of Procyte dot plots. Next, we'll look at some typical dot plot patterns that you'll find in the normal practice of veterinary medicine.